Wow, this week in longevity biotech research has been really something else. We've got a ton to cover today.
Yeah, there's been a deluge of new papers, some really pushing the boundaries.
Absolutely. So today we're diving deep into 5 papers. These were all fresh off the press, published just this week. What makes these stand out is that they come from really credible institutions, and the findings are, well, surprising, to say the least. The kind of stuff that might force us to rethink some of our models of aging.
Exactly. We're not talking about small incremental steps here. This is research that has the potential to really shift paradigms.
And for you, the listener, that's what we're here to unpack today. We want to distill the key insights, figure out why they could be so impactful in the field of longevity, and of course, always stay skeptical.
Right. A healthy dose of critical thinking is essential.
Exactly. We need to be able to separate the truly novel from the, well, more incremental advances. And to keep you oriented, we're looking at papers published this week starting on Saturday, March 22nd, 2025, and going all the way to today, Thursday, the 27th.
Sounds good. Let's dive in.
Okay. First up, a paper that came out on Wednesday, March 26th. It's called Bisphosphonates Trigger Anti-Aging Effects Across Multiple Cell Types and Protect Against Senescence. The authors are Liu, Rao, Knowles, and a bunch of others. They're from the Botnar Research Center at the University of Oxford. And you can find this one on biorxiv. Now, you might have heard of bisphosphonates before.
Yeah, they've been used to treat bone loss disorders for quite some time.
Right. For decades, really. We've mainly thought of them as, helping maintain bone health. But more and more, it's looking like these compounds might have benefits that go way beyond the skeleton.
I think we're starting to see hints that they could be linked to, reduced all-cause mortality and even lower rates of some age-related conditions.
Which is what makes this research so surprising. The Oxford team discovered that in non-skeletal cells, meaning cells that aren't part of our bones, bisphosphonates seem to preferentially hang out with lysosomes and endosomes.
So essentially the cell's recycling and waste disposal units.
Yeah, exactly. But even more unexpected, they found that at low doses, these drugs actually stimulated cell growth. Now, here's where it gets really interesting. They used a technique called in vivo spatial transcriptomic analysis on aged mice.
Ah, that's a pretty sophisticated method.
Yeah.
Lets you see which genes are active in very specific locations within a tissue.
Right. It paints a much more detailed picture than traditional methods. And what they found in the mice treated with bisphosphonates was a decrease in markers of senescence across multiple organs.
And just to clarify for listeners who might not be familiar, senescence is basically when cells stop dividing and start releasing all sorts of inflammatory signals, which contributes to aging.
Right. And not only that, but the cellular makeup of these organs actually shifted towards what you'd normally see in younger mice. So the big takeaway here is that a common bone drug might actually have broader anti-aging effects by, interacting with these cellular recycling centers and reducing the number of these aged senescent cells.
It's a very intriguing finding. And they didn't stop there. They then took a look at human data. They analyzed the plasma proteome of patients with osteopenia before and after treatment with bisphosphonates.
Now, for anyone who's not sure what that means, the plasma proteome is essentially the entire collection of proteins that are circulating in your blood.
Right. It's a snapshot of what's happening in the body at a molecular level.
Exactly. And they found significant changes in around 400 of these proteins after bisphosphonate treatment. But these weren't just random changes. These included things like regulators of GTpases, which are these crucial signaling molecules involved in loads of cellular processes.
And also markers of key aging mechanisms, not just senescence itself, but also autophagy, apoptosis, and inflammation.
Yeah. Pretty wide range of effects. And to add to that, their in vitro work showed that treating cells with bisphosphonates actually protected them from becoming senescent in the first place.
Really interesting. It suggests that these drugs might be acting at multiple levels to counteract the aging process.
Right. They then did a really deep dive into the data and were actually able to identify some new protein targets that bisphosphonates seem to bind to directly. These are called PHB2 and ASAH1. And by combining all of their data, the RNA sequencing, the ATAC sequencing, which looks at DNA accessibility, and the patient data, they found evidence suggesting that a transcription factor called MF2A might be regulated downstream in the heart.
So essentially, they're piecing together the molecular puzzle of how bisphosphonates might be working.
Exactly. So it seems like these drugs might be having these unexpected benefits by directly interacting with non-skeletal cells through these newly discovered protein targets and by fundamentally changing the protein landscape in our blood.
Yeah, and that could have some pretty significant implications for pathways linked to both cellular senescence and the broader aging process.
Absolutely. In terms of longevity relevance, this feels potentially huge. This is a class of drugs with a long history of use, so we already know a lot about their safety profile.
Right, which is a big advantage when it comes to developing new therapies.
Exactly. And it looks like they might be hitting some of the core drivers of aging, like senescent cells and these protein imbalances. Plus, the fact that they're already approved for human use means that the path to translating this into actual therapies could be a lot shorter.
I agree. It's definitely worth exploring further. And it also makes you think about previous research connecting lysosomal health to aging, perhaps even linking back to pathways involving proteins like SIRT1.
That's a great point. There are definitely some interesting connections to explore there. But of course, we also need to be cautious.
Absolutely. I mean, the human data they presented was all from people with osteopenia. So does that mean we can generalize these findings to people without bone loss?
Yeah, that's a key question. And while they found these new protein targets, the exact mechanisms of how bisphosphonates interact with lysosomes and then influence senescence, well, that's still not entirely clear.
Right. And we can't ignore the potential long-term risks associated with bisphosphonates either, like osteonecrosis of the jaw. We really need to weigh those risks against the potential anti-aging benefits, especially if we're talking about using these drugs in a broader population.
For sure. Okay, let's move on to our second paper. This one also came out on Wednesday, March 26th. It's titled Get Ready for It, Targeted Intervention of Senescence, induced by a 5.5 fragment of tRNASec NCA with antisense oligonucleotide extends healthspan and lifespan in mice.
Wow, that's a mouthful.
I know, right? The lead authors are Cao, Bai, and Zhang, and they're from the State Key Laboratory of Quality Research in Chinese Medicine at Macau University of Science and Technology. And you can also find this one on biorxiv. This research dives into the role of damage transfer RNA, or tRNA, in aging.
Okay, so we know that a big focus in longevity research is identifying the fundamental hallmark of aging and developing therapies that target them directly.
Right, that's kind of the holy grail.
It's exactly. But figuring out the real drivers, especially within the super complex changes that happen in our epigenome, that's proving to be a real challenge.
And the epigenome, just to remind everyone, is that layer of regulation that sits on top of our DNA, controlling which genes are turned on and off.
Right. It's like the software that runs on the hardware of our DNA.
Perfect analogy. So this team has made a really interesting discovery. They found that a particular type of tRNA called tRNASec, NCA, seems to be the most severely damaged tRNA species in the kidneys of aged mice. But it's not just that the damage itself is a marker of age. They found that this damage messes up the regulation of selenoproteins.
And those are a group of proteins with some pretty potent antioxidant and anti-aging effects.
Right. And to make things even more intriguing, this damage results in the creation of this specific fragment, a 5' tRNA half, that actually builds up as mice age. So the key insight here is that this damage to a specific RNA molecule seems to be more than just a side effect of aging. It's producing a byproduct that might actually be driving the aging process itself.
It's like a double whammy. And what's really fascinating is the mechanism they uncovered. This 5' fragment of tRNA-seq NCA actually interacts with and activates toll-like receptor 7, or TLR7.
Now, I know TLR7 is a key player in our innate immune system, our body's first line of defense, but how does that tie into aging?
Well, activating TLR7 triggers immune responses, and crucially, it also promotes cellular senescence in both mouse and human cells. So what this research is doing is directly linking damage to this particular RNA molecule with inflammation and aging, this concept we often call inflammaging.
Right, that idea that chronic low-grade inflammation is a key driver of age-related decline. So it sounds like they've found a potential new pathway that's contributing to the whole aging process.
That's exactly right. It's a novel connection. And the therapeutic approach they took is really exciting. They gave aged mice an antisense oligonucleotide, or ASO.
Now, I always think of an ASO as kind of a molecular ninja, sneaking in and specifically targeting a rogue RNA molecule. Is that a Decent analogy?
Yeah, that's a good way to think about it. It's a very precise tool.
So in this case, their ASO was designed to target that 5' tRNASec NCA half. And the results were pretty amazing. They saw improvements in a bunch of aging markers, an increase in telomere length, those protective caps on the ends of our chromosomes.
The ones that tend to shorten with age.
Exactly. And get this, they even saw an extension of both health span and lifespan in these aged mice.
Wow, that's significant. So it suggests that if we can target this one specific damaged RNA fragment, we might be able to have these broad anti-aging effects.
So this paper is really suggesting that tRNA damage isn't just a passive bystander in aging, but it might actually be a brand new hallmark of aging that we can actively target for therapeutic benefit.
Absolutely. And to take it a step further, they discovered that this ASO also plays another role. It directly targets and downregulates BCAT1, an enzyme that's involved in the metabolism of branched-chain amino acids. And it does this through a process called RNA interference, or RNAi. So basically, this ASO is multitasking, tackling senescence from multiple angles. This paper is a big deal because it provides the first direct evidence I've seen of specific tRNA fragments acting as actionable aging hallmarks. It really bridges the fields of tRNA biology and inflammation.
Wow, that's fascinating. And it definitely makes you think about all that other research exploring the role of inflammatory signals in aging, like the work on GDF15.
Exactly. It all ties together. But, as always, there are some caveats. TLR7 is a critical part of our immune response, especially when it comes to fighting off viral infections.
So if we're messing with its activation, couldn't that have unintended consequences for our immune system?
That's definitely something to consider. We need to understand if there are any potential downsides to targeting this pathway. And the paper doesn't go into much detail about the tissue specificity of these effects either.
So is this mainly a problem in the kidneys, or are we seeing similar things happening in other organs?
That's another key question that needs further research. And as with any ASO-based therapy, we have to be mindful of potential off-target effects. Could the ASO be binding to other RNA molecules and causing unintended problems? It's worth mentioning that there's been previous work on the role of tRNA fragments in aging, including some interesting findings that came out of Harvard back in 2023.
So this new research builds on that foundation, but provides a much deeper understanding of the mechanism and gives us a more clear therapeutic target.
Exactly. Now for something completely different.
Right, let's move on to our third paper. This one was published a day earlier on Tuesday, March 25th in the journal Nature Aging. It's titled Epigenetic Editing at Individual Age-Associated CPGs Affects the Genome-Wide Epigenetic Aging Landscape. Bit of a mouthful, but it's a fascinating one. Lead author is Sven Liesenfelder from the Institute for Stem Cell Biology at RWTH Aachen University Medical School in Germany.
This paper really gets into the nitty-gritty of epigenetic clocks, which is such a hot topic in aging research.
And just a quick reminder for anyone who's not familiar, epigenetic clocks are those really accurate predictors of biological age based on changes in DNA methylation patterns.
Right. So this team took a super interesting approach. They used CRISPR-guided epigenetic editing to target very specific CPG sites.
Now, I always struggle to remember what CPG sites are. Could you give us a quick refresher?
Sure. So CPG sites are regions in our DNA where a cytosine nucleotide is followed by a guanine nucleotide. And these sites are really important because they're often targets for methylation, which can change how genes are expressed.
Ah, okay. So they used CRISPR to basically manipulate these methylation marks at specific sites that are known to change with age.
Exactly. And here's the really surprising thing. They found that by targeting just a single age-related CPG site, they actually triggered changes in methylation at other age-associated regions across the entire genome.
So it's like a domino effect.
Kind of. They called it genome-wide bystander effects. And these effects were really consistent and reproducible.
So it's suggesting that the way our epigenome ages is much more interconnected than we thought. Like if you tweak one part of it, it can have these ripple effects throughout the whole system.
That's a great way to put it. And to really dig into this interconnectedness, they used a technique called 4C sequencing.
Which, for those of us who aren't molecular biologists, means
It lets researchers see how different regions of the genome are physically interacting with each other in three-dimensional space.
Ah, okay. So it's not just about the linear sequence of DNA, but how it's all folded and organized within the cell.
Exactly. And what they found was increased interactions between the edited sites, the regions where they saw these bystander modifications, and other age-related CPG sites. It really points to this idea of a whole network of coordinated epigenetic changes that happen as we age.
Fascinating. So it's not just random changes, it's like a carefully orchestrated process.
That seemed to be the case. And they took this even further and did multiplexed epigenetic editing in human T cells and mesenchymal stromal cells, two really important cell types in the body. They targeted 5 different genomic regions that either gain methylation with age, which is called hypermethylation, or lose methylation with age, which is hypomethylation.
So they were basically trying to mimic what happens naturally during aging, but in a much more controlled way.
Right. And interestingly, they found that targeted methylation was more stable at those sites that naturally become hypermethylated with age.
Yeah.
And what's really important is that both approaches, whether they were targeting sites of increased or decreased methylation, ended up causing these bystander modifications at CPG sites that have the strongest correlation with chronological age. So it's impacting both the gains and losses of methylation that we typically see with aging.
Okay, so what does all of this mean for longevity research?
Well, it provides some pretty convincing evidence that we can actually manipulate this whole epigenetic aging network by just targeting a few key sites.
Hmm.
It strongly suggests that there's a causal link between epigenetic changes and aging, meaning that these changes aren't just a passive result of getting older, but might actually be driving the aging process itself, which in turn means that we might be able to reverse or at least slow down aging by targeting the epigenome. This builds on the groundbreaking work of Steve Horvath and his development of epigenetic clocks, but it starts to give us a much clearer understanding of the mechanism behind these changes. It also ties into all the exciting work being done on partial cellular reprogramming at places like Altos Labs. The idea is that we might be able to reset the age of our cells by manipulating the epigenome.
So it's a really exciting concept, but like with all of these new discoveries, there are still lots of questions.
Oh, absolutely. We still need to figure out the functional relevance of these methylation changes.
Hmm.
Just because we can change them doesn't automatically mean we're improving cellular function or overall health.
Yeah.
And then there's the huge challenge of developing safe and effective ways to deliver these epigenetic editing tools to the right cells and tissues in a living organism.
Right, that's always a major hurdle when it comes to translating these lab-based discoveries into actual therapies.
Exactly. And we also can't forget about the potential risks of those bystander effects. When we're making these widespread changes to the epigenome, could we be accidentally disrupting something important, like genes that protect against cancer or other crucial regulatory elements?
A really important point. We need to make sure that these widespread changes are actually beneficial and that we're not inadvertently causing harm.
Agreed. Moving on.
Okay, let's switch gears and talk about a paper that came out a bit earlier in the week on Saturday, March 22nd. This one is titled Exosomes Released from Senescent Cells and Circulatory Exosomes Isolated from Human Plasma Reveal Aging-Associated Proteomic and Lipid Signatures. The authors are Patel, Bonds, Rose, and a bunch of other researchers from the Buck Institute for Research on Aging. You can find this one on biorxiv. Now, this research zeroes in on the role of cellular senescence in aging. And again, just a quick reminder, senescence is when cells enter the state where they stop dividing and start releasing all sorts of inflammatory molecules.
Right, it's like they become these little factories churning out pro-aging signals.
Exactly. And these senescent cells release this whole cocktail of molecules called the senescence-associated secretory phenotype, or SAP, which includes these tiny vesicles called exosomes.
Ah, exosomes, those tiny packages that cells use to communicate with each other.
Yeah, exactly. And they can travel to other tissues and potentially spread those senescent signals far and wide.
Like little messengers of aging?
So the team at the Buck Institute did this really in-depth analysis of the protein and lipid content of these exosomes. They looked at exosomes released by senescent human lung fibroblasts that they were growing in the lab, and they also looked at exosomes that they isolated from the blood plasma of both young and older people. And they used some pretty sophisticated mass spectrometry techniques for this.
So was it really comprehensive analysis?
Yeah, they weren't messing around. And they uncovered a ton of information. In the exosomes released by those senescent cells in the lab, they found around 1,300 different proteins.
Wow, that's a lot of data.
I know, right? And when they looked at the exosomes from the human plasma, they found over 1,350 proteins, with 171 of those showing significant differences in abundance between the young and old groups. But here's the really interesting part. 52 of the proteins that were different in the plasma exosomes of older people were also present in the exosomes released by those senescent fibroblasts in the lab.
So there's this overlap between what the senescent cells are pumping out in the lab and what's actually circulating in the blood of older people.
Exactly. It really suggests that those senescent cells are playing a role in shaping the composition of those circulating exosomes as we age. And the overlap included some interesting characters like serine protease inhibitors, prothrombin, coagulation factor V, plasminogen, and reelin.
All involved in important processes like blood clotting and the structure of the brain, among other things.
Right. They also did a lipidomic analysis looking at the fats within the exosomes, and they found 247 different lipids. And they saw that after they induced senescence in those cells in the lab, there were significant increases in certain types of lipids: phosphatidylcholines, phosphatidylcholine, and sphingomyelins. So this indicates that the membrane composition of these exosomes changes when cells become senescent.
Right, so it's not just the protein cargo that's important, but also the lipid makeup of these little packages.
And they also pointed out that the proteins that changed the most were involved in things like remodeling of the extracellular matrix, which is that scaffolding around our cells, and inflammation, both of which are heavily implicated in aging.
Yeah, it all fits together. And this research aligns really well with what companies like Unity Biotechnology are doing, developing drugs to selectively eliminate senescent cells, or senolytics.
Right. The idea is to clear out those senescent cells before they can cause too much trouble. So this paper is kind of like the first really comprehensive multi-omics map of the protein and lipid contents of these senescence-associated exosomes. And what's the potential impact of all this for longevity research?
It gives us valuable insights into the mechanisms of how these senescent cells might be communicating with the rest of the body and contributing to aging at a molecular level. And the specific protein and lipid signatures that they identified in these exosomes could potentially be used as biomarkers.
So we can maybe measure those in people to get an idea of their senescent cell burden.
Exactly. And those specific molecules could also be potential targets for therapies aimed at reducing the harmful effects of senescence.
But as always, we have to be careful about jumping to conclusions about cause and effect.
Right. Are these exosome signatures actually driving aging, or are they just a consequence of it? We don't know for sure yet.
And we also need more research to confirm that these findings hold true in larger groups of people and to see if these exosome contents actually correlate with real-world measures of health and aging.
Definitely. And technically speaking, isolating those exosomes from blood plasma can be a bit tricky, so we also need to be aware of potential technical artifacts that could be influencing the results.
Good point. So lots more research to be done, but this is a really promising area. Okay, ready for our final paper?
Hit me with it.
All right. This one was published on Wednesday, March 26th, and it's called SIRT6 Activator Fucoidan Extends Health Span and Lifespan in Aged Wild-Type Mice. The authors are Biashad, Hillpot, Morandini, and their colleagues at the University of Rochester. You can find this one on bioRxiv as well. This research focuses on the potential of activating a key longevity-related protein, and you've probably heard of this protein before. It's called SIRT6.
That's SIRT6,
Yeah.
a classic. We know it plays a vital role in regulating several pathways that are really important for longevity, like DNA repair and the silencing of line-1 retrotransposons.
And those are?
Those are repetitive DNA sequences that can become active as we age and cause all sorts of genomic instability. It's like they're jumping genes that can wreak havoc on our DNA.
Yikes, that doesn't sound good.
Not good at all. And we know that mice that are genetically engineered to lack SIRT6 die prematurely. But studies have shown that boosting SIRT6 levels can actually extend lifespan, at least in male mice.
So it's a pretty attractive target for longevity interventions.
Absolutely. And this research team has been investigating fucoidan, which is this complex sugar molecule found in brown seaweed.
Seaweed? Really?
Yeah, you wouldn't think it, but seaweed is actually a treasure trove of bioactive compounds. And previous research had suggested that fucoidan could activate one of SIRT6's main functions, its decetylase activity.
And just to clarify, decetylation is...
It's a process that involves removing acetyl groups from proteins, and that can have a huge impact on how those proteins function.
Got it. So fucoidan was already looking promising as a potential SIRT6 activator.
Right. But now they've made a new and pretty exciting discovery. It seems that fucoidan also activates another important enzymatic activity of SIRT6. It's mono-ADP ribosylase, or HAV-ADPR activity.
Say that 10 times fast.
I know, right? But the cool thing is that some studies have found that this MADPR activity is actually elevated in human centenarians, those folks who live to be over 100 years old.
Ah, so this is starting to sound even more promising. So this one natural compound might be able to boost multiple beneficial functions of a key longevity protein.
Exactly. And it gets even better. They found that giving fucoidan to aged male mice significantly increased their median lifespan.
Mm.
But they also saw positive effects in both male and female mice, including a marked reduction in frailty, which is basically a measure of overall health and resilience in older individuals.
So they were healthier and lived longer. That's a pretty good combo.
It is. And they also saw a decrease in their epigenetic age, as measured by those DNA methylation patterns.
So biologically speaking, they were younger.
In a way, yes. And they did some further digging to figure out how fucoidan might be working its magic. They found that in the mice treated with fucoidan, there was a suppression of line 1 elements, those jumping genes we talked about earlier.
So it looks like RT6 might be playing a role here, helping to keep those pesky retrotransposons in check.
Exactly. And that actually aligns with some of the work that David Sinclair has been doing, highlighting the role of retrotransposon activation in aging. It's also interesting to note that brown seaweed, which is rich in fucoidan, is a common part of the diet in countries like South Korea and Japan.
Which, as you might know, have some of the highest life expectancies in the world.
Right. So it's tempting to speculate that maybe there's a connection there. Maybe fucoidan supplementation could be a safe and relatively easy way to activate SRET6 and potentially improve human health span and lifespan.
It's a really intriguing possibility, but of course we also need to be a bit cautious here.
Always. One big question is the bioavailability of orally administered fucoidan. How much of it actually gets absorbed from our gut and reaches the tissues where it needs to work? We don't really know.
Yeah, that's a crucial question. And they only saw the lifespan extension in male mice. We don't yet know why there's this sex-specific effect.
Right. And as they mentioned in the paper, there could be other beneficial compounds in those seaweed-rich diets that are contributing to the longevity seen in those populations. So we can't say for sure that it's all due to fucoidan.
So more research is definitely needed to tease apart these effects. All right, we've covered a ton of ground today, looking at 5 really exciting papers that just came out this week. What are some of the big themes that stand out to you from all of this?
I think the main takeaway is that there's a lot of really innovative research happening right now in the field of longevity. We're seeing new ways to target cellular senescence, not just by getting rid of those aged cells directly, but also by potentially repurposing existing drugs like bisphosphonates, which seem to have these unexpected anti-aging effects.
Like giving those old drugs a new lease on life.
Exactly. And the field of epigenetic reprogramming is moving forward rapidly, both directly editing specific CPG sites and activating key regulatory proteins like RT6 are showing real promise in terms of influencing the epigenetic landscape of aging. And maybe the most exciting thing is that we're uncovering new and potentially fundamental hallmarks of aging, like those damaged tRNA fragments and the specific lipid signatures in those exosomes. This is really expanding our understanding of the aging process at its core and opening up whole new avenues for therapeutic intervention.
And it's amazing how some of these potentially huge findings are coming from unexpected places, like the potential anti-aging effects of a drug that's been used to treat bone health for decades.
I know, right? It just goes to show that we need to keep an open mind and be willing to look beyond the traditional uses of drugs and compounds. There might be hidden gems out there waiting to be discovered.
Absolutely. But it's also crucial to remember that all of this research is still in its early stages. These are exciting initial findings, but they all need much more investigation, especially when it comes to confirming these results in humans and figuring out exactly how these mechanisms work.
Couldn't agree more. We need well-designed clinical trials in people and more detailed mechanistic studies, both in animal models and in the lab, to really validate these findings and make sure that any potential therapies that come out of this research are both safe and effective.
So for you, our listener, I want to leave you with a couple of questions to ponder. We've talked about all these different approaches, from repurposing existing drugs to targeting specific RNA fragments, manipulating the epigenome, and even exploring natural compounds. Which of these avenues do you think holds the most promise for extending healthy human lifespan in the coming years? And what crucial questions still need to be answered before we can truly turn these exciting scientific discoveries into real-world benefits for human health and longevity? We encourage you to dive deeper into these papers yourself and to stay curious about the ever-evolving and fascinating field of longevity research. Thanks for joining us on this deep dive.
Thanks, everyone.